Hi, I'm Gia.
And I'm Sean.
Our project is an analysis of the crystal structure of UmaA from Mycobacterium tuberculosis complex with S-adenosylmethionine-dependent or SAH. So first we'll introduce M. tuberculosis and the role of UmaA in the metabolic pathway. Then we'll get into the structure, interactions, and alignments that we did to better understand the structure. Then we'll talk about a potential inhibitor that we found. So Mycobacterium tuberculosis is a gram-positive bacterium that causes tuberculosis, or TB, as you might know, and it is the leading cause of death worldwide. It kills about 1.8 million people each year, especially in developing countries. And while many of the drugs that were developed in the 1950s are effective against a large percentage of TB cases, there is a growing threat of drug resistance as the microbes adapt to survive in the presence of first-line and even second-line drugs. So it's very important to continue studying this to find new targets and develop new treatments. This bacterium is very virulent and persistent, in part because of the cell wall, which is made up of mycolic acid, and modifications to these mycolic acids can result in resistance to chemical injury and low permeability to antibiotics.
So one thing that aids in the survival of M. tuberculosis is the modification of mycolic acids. And there is a batch of S-adenosylmethionine-methyltransferases known as cyclopropane mycolic acid synthases, or CMASs, that is responsible for making these modifications. These include CMAA 1 through 2, MMAA 1 through 4, PCAA, and UmaA. Modifications can vary depending on the enzyme. For example, you can have the addition of cyclopropyls, methyl branches, hydroxyl groups, and methoxy groups. As shown in the image, all the functions of the CMAS family members are mentioned except for UmaA, and that's because UmaA is the least known of the CMAS enzymes. So the modifications of mycolic acids is just a small piece of the complicated metabolic pathway of mycolic acid synthesis. As shown, there are many drugs that have been approved to treat tuberculosis, and they interfere at many intermediary steps in this pathway. There are currently no drugs for the modifications of mycolic acids, and that is where we come in. So CMAS enzymes make modifications to mycolic acids using S-adenosylmethionine, or SAM, as a methylating agent. SAM, on the left, has an electrophilic methyl group due to its electronegative sulfur atom. So a nucleophile can attack the electrophilic methyl group, thus making the nucleophile methylated and SAM being demethylated to form S-adenosyl homocysteine, or SAH. And SAH is what is complex to our protein structure, UmaA. So this is an overview of UmaA, which was determined by X-ray crystallography by the Seattle Structural Genomics Center for Infectious Disease. UmaA has two domains. The SAM binding domain is shown in blue and consists of a Rossmann-like fold, and the substrate binding domain is shown in yellow and consists of a helical lid. The mycolic acid binds to the substrate binding domain. There is also a GX-GXG motif shown in magenta, which is the most conserved sequence in UmaA. So SAH, the product of the reaction, as stated before, is in our structure. PEG6 and NO3- are not the natural ligands, but are reagents for crystallization added by the crystallographers that happen to interact at these locations on the protein. PEG6 is a long chain of ether functional groups, so this is clearly industrially synthesized. It does mimic the long alkyl chain, which is why it would make sense for it to bind to the mycolic acid binding site. NO3- was also added by the crystallographers and mimics the naturally occurring molecule HCO3-, or bicarbonate. HCO3- acts as a general base for this reaction, so one can easily imagine that NO3- takes the place of HCO3- due to its trigonal planar structure.
So we use Dolly, which is a web server using distance matrix alignment to compare structures to determine and align structural homologs for UmaA. So root mean squared deviation, or RMSD, which is a measure of the average distance between atoms, was used as a guideline to find structures that best fit with our protein. These included proteins from different bacteria, archaean, a parasite, plant, rat, and a human. And the diversity of species containing homologs of our protein UmaA reflects the versatility of the SAM methyltransferase structure. So you can see towards the bottom, that the structure of the SAM binding region with the Rossmann-like fold is pretty highly conserved among these varying species. But actually, despite the similarity in structure and the fact that each enzyme binds at the equivalent position of the fold, the residues there are generally not conserved except in this region, which is the most highly conserved in all these methyltransferases. On the other hand, the auxiliary N-terminus region is a lot more varied, and this may be because of the variety of different substrates that need to be methylated by the region.
So this is the interaction of SAH with UmaA. Each component of SAH, the methionine, the ribose, and the adenine ring, is interacting with specific residues. The glycine-rich region has glycine residues that also interact with the ribose moiety in the SAH. Here's a superimposed image of NO3- and HCO3- taken from related proteins. As shown before, since both molecules are trigonal planar, they will be located in the same place and overlap well. The amino acid residues interacting with NO3- are glutamate, histidine, cysteine, and tyrosine. What's important to note is that all these residues are able to engage in general acid-base chemistry and further supports the hypothesis that HCO3- acts as a general base for the catalytic reaction.
And here we have PEG6, which is one of the ligands within our crystal structure that was captured during the crystallography process. And we basically superimposed it with a homolog, which contained EPH, a more natural phospholipid. So EPH is interacting with a lot of the hydrophobic residues shown in orange. And you can see that it pretty much traces well with the PEG6 until it reaches this point where it makes a U-turn while the PEG6 keeps going. The PEG6 is long and stringy, similar to a phospholipid, so it might need a long type of pocket to accommodate this type of structure. This is not the natural substrate. It's very polar compared to EPH, so it might bind in an unusual way. We think that the ether groups might allow it to have a different conformation.
Here we depict how the reaction might proceed. Based on crystal structures taken from the protein data bank, we made a spatial model which is shown on the left. Based on the structure of SAH, we created SAM by building a methyl group onto the sulfur of SAH. We also replaced the unnatural ligands added by the crystallographers with more natural ones. So we superimposed HCO3- onto the structure and removed NO3- and superimposed EPH onto the structure and removed PEG6. Based on the positioning of the molecules and previous experiments, we made inferences for a mechanism. I must emphasize that the general mechanism for UmaA is not known. In fact, as stated before, we do not even know what UmaA produces. One mechanism could be that the alkene will perform a nucleophilic attack on the electrophilic methyl group of SAM and form SAH and a carbocation intermediate. The carbocation intermediate is stabilized by tyrosine 32 located above it, and the bicarbonate ion acting as a general base will deprotonate the added methyl group on the carbocation intermediate and cause the methyl group to collapse inward to form a cyclopropane ring.
So if you recall, there are several paralogs of UmaA, meaning that they're from the same organism but with different functions. In this case, they're all part of the CMAS family, responsible for modifying mycolic acids as described earlier. So a structural alignment was done with those that we could find on PDB to examine variable regions in the helical grid domain. So this is a view of the N-terminus, and again, you see that this region is very variable. And the UmaA in yellow and the MMA4 are in purple are the only two which have the less defined N-termini that don't close on the mycolic acid binding site. We found this interesting, so we investigated it further using AlphaFold.
So we decided to do a comparison of the N-terminus between the structure from X-ray crystallography and the predicted structure from AlphaFold. And AlphaFold is an AI software that's able to predict protein structure based on their amino acid sequence. So looking at the crystal structure, we see that the electron density in the N-terminus is quite broken. For example, we see glutamine, glutamate, and phenylalanine residues. These residues are supposed to have large side chains, but they're clearly missing. This suggests that the structure is very flexible. In addition, the predicted structure from AlphaFold deviates significantly from the crystal structure. Whereas the N-terminus crystal structure protrudes outwards into the solvent, the predicted N-terminus structure from AlphaFold is closed onto the active site. However, the AlphaFold predicts this structure with very low confidence, which further suggests that the N-terminus is not well defined. One reason for this may be that the N-terminus acts as a molecular gate, where it can be switched from an open state to a closed state. In its open state, it can allow for substrates to enter, and in its closed state, it can allow for the catalytic reaction to occur.
So given the role of UmaA in the metabolic pathway of mycolic acid synthesis, the identification of potential inhibitors for UmaA and related enzymes is a widely explored topic. One analog of SAH containing different alkyl groups was studied as a potential inhibitor for the reaction, and this is SADE. So SADE was identified because it was found from previous experiments to be a very effective inhibitor of an ortholog in E. coli and an even more potent inhibitor for mycolic acid methyltransferases. So again, we superimpose the PDB structure containing SADE onto our structure, and you can see that the structural similarities between SADE and SAH may allow it to interact with the same residues. And this alkyl chain for SADE even spans into the hydrophobic pocket, where we predict that it might be able to interact with the nonpolar residues via hydrophobic interactions. So this would essentially allow it to potentially bind to two sites at once, which makes it a very good potential inhibitor just based on the structure. This is obviously not tested in the lab yet. It's just a hypothesis based on literature searches and structural analysis.
So from these conclusions, we hope in the future that we can perform protein dynamics studies and in vivo and in vitro studies to further test and confirm our hypothesis to aid in the identification of novel drugs and targets for tuberculosis.
We want to thank Dr. Craig Smith, UMA, and Heiko for advising and supporting us throughout the project, as well as those from the SSGCID for providing the crystal structure for UmaA. Thank you.